first we start with sample preparation. So first we extract protein from cell or tissue using a special buffer. Then measure the protein concentration to make sure the same amount of protein is loaded into the gel for consistent result. Next is gel electrophoresis, which is also known as STS page. The protein are mixed with a detergent called STS, which is unfold them and give them a negative charge. We apply an electric current, the proteins start moving through the gel. So the next step is transfer to membrane, which is we transfer those separated protein into a membrane like nitrocellulose or PVDF sheet. So the fourth step is blocking. To make sure the antibodies only stick to the target protein, we block the membrane with a solution like milk or PSA.
The next step in Western blotting is primary antibody incubation, where a specific antibody binds to the target protein. Then we add a secondary antibody that binds to the primary and amplifies the signal. This secondary antibody is tagged with an enzyme like HRP, which reacts with a detection substrate to produce light or color. The brighter the signal, the more protein is present. Finally, we analyze the results by comparing protein bands to a molecular weight marker and housekeeping proteins for accurate quantification. Western blotting is essential for detecting specific proteins in complex samples.
So for the next part, we'll be explaining about the important role of antibodies in the Western blotting process. Antibodies play an important role in the Western blotting technique because it used to detect specific proteins in the sample. For example, we have the first one, which is the primary antibodies are used to bind to the specific protein of interest. They bind directly to the target protein on the membrane after electrophoresis and transfer. Primary antibodies can either be polyclonal, which recognizing multiple epitopes on the target protein, or monoclonal, which recognizing a single epitope. So the next antibodies is secondary antibodies. They are used to detect primary antibodies and they are typically conjugated with enzymes or fluorescent dyes. The secondary antibodies bind to the primary antibodies, amplifying the signal for detection. So, moving on is loading control. These antibodies are used to ensure accurate quantification by normalizing protein loading across different samples.
Furthermore, Western blotting also commonly used in laboratory, especially in scientific research. This technique can be applied to many important usages. For example, it can detect circulation of antibodies specific to a single protein or several proteins. There is a research that has been done to observe the production of a monoclonal antibody specific to a sablefish IgM. In this research paper, Western blotting used monoclonal antibodies to detect sablefish IgM, identifying specific proteins and immunogenic regions with high precision. Other than that, this technique can also analyze biomarkers such as hormones, growth factors, and cytokinase. There are also research about platelet protein biomarker panel for ovarian cancer diagnosis. Western blotting is used to identify biomarker protein and expression in platelets using specific primary and secondary antibodies for selective detection. Lastly, this technique can also be used for analyze gene expression studies. A research also has been done on profiling protein expression in circulating tumor cells using Western blotting. This technique is used to analyze protein expression in CTCs by using antibodies to detect specific target proteins, enabling precise cancer marker profiling.